I am Dr. Samantha Levell, and I am an assistant professor of marine biology here at New College of Florida. We have a really cool opportunity as faculty here to create these immersive group ISPs, and the objective of these ISPs is for generally first-year students to sort of get to understand what an independent study project is and how they can get involved in more research and more opportunities that could potentially lead them into their thesis project. And so for this ISP in particular, I was thinking about ways that I could get a group of students together and really focus on learning lab skills and lab techniques so that they would feel really comfortable with those things going forward into their classes and also their potential future research. The students face challenges in sort of all aspects of this ISP, and one of the main reasons for that is that this type of protocol is really specialized. So this is what's called an enzyme-linked immunosorbent assay. Dissections are hard. Sharks are really tough, right? They have their dermal denticles on their skin. Their skin is basically covered in tiny teeth. It requires some pretty precise yet firm dissection tools and identifying anatomical features. So there were a lot of things along the way that basically everything would be a new challenge for the students. But I've both been really impressed with their ability to tackle these new challenges and also create work to create an environment in which everybody feels comfortable with the next step.
Hi, my name is Gigi Tan, and I am a third-year marine biology student. This ISP about Carcharhinus limbatus, the blacktip sharks, is literally what I want to do with my entire life. Ever since I was 6 years old, I've always wanted to be a shark biologist. And a lot of colleges don't do hands-on experiences, or they don't spend a full month just doing one full class and diving super deep and intensely into one topic. And I think that's super valuable.
Well, one of the things that was unique here is that we were looking for brain tissue because we're looking at Brevetoxin, which is a toxin, a neurotoxin produced by Carcharhinus brevis, which is a harmful algae in the area. And we were looking at Brevetoxin concentrations in the brains, livers, muscles, stomach, and gills of the sharks. So not only are they getting to identify and learn about what each of those tissues does, but they're also doing an incredibly intricate dissection in which they need to actually break through the cranium of the shark to access that brain tissue. So they're learning about the anatomy of a shark in a much more in-depth way than they would from a sort of typical shark dissection.
So being at two institutions now, I've never had as close a relationship with my faculty as I've had at New College, and that's really helped me to grow and become a better biologist.